I would like to discuss Anfinsen's thermodynamic model of protein folding. So before coming to Anfinsen's experiment, I would like to explain the significance of protein folding. As we know that every protein folds into a three-dimensional structure which is compact and biologically active. This uniquely folded structure is also known as native conformation, which is commonly used term in biochemistry. So native conformation simply means that the protein is active, it is folded and biologically active. The area of protein folding is also important to understand molecular pathogenesis of disease. There are many diseases which are caused due to defect in protein folding, such as Alzheimer's, Parkinson's, Huntington, and cystic fibrosis, and many more. So why do proteins undergo folding? I will explain this in relation to thermodynamic principles. According to thermodynamics, a protein molecule folds to acquire the most stable state, which is characterized by the least energetic and highest entropic state. Therefore, you can say that the folding is driven by the principles of thermodynamics involving minimization of Gibbs free energy. Now I would like to explain Gibbs free energy and its relationship with other thermodynamic parameters with the following equation: delta G is equal to delta H minus T delta S, where delta refers to the energy difference between the folded and unfolded structures. G is Gibbs free energy, or you can say the usable form of energy. H is enthalpy or total energy content of the molecule. S is entropy or measure of disorderliness, whereas T is absolute temperature in degree Kelvin. For a spontaneous reaction, delta G must be less than 0 kilocalorie per mole. In fact, for a folded protein, delta G lies between -5 to minus 20 kilocalorie per mole. It means that the protein under normal physiological condition, a folded protein is not remarkably stable. Therefore, we can say that a folded protein is only marginally stable. However, this is important fundamentally for providing metabolic flexibility and for allowing regulation of enzyme activity with slight change in the energy at physiological condition. So coming to Anfinsen's experiment, it is important that you understand the components he used in his experiment. They are RNAs, which was used to study protein folding. Second was urea, and the third one is the beta mercaptoethanol. So I will like to go over the characteristics of these components. The first one is RNase, which is basically an enzyme that hydrolyzes RNA molecule. It contains 124 amino acids, including 8 cysteine residues. This is important to note that these 8 cysteine residues can make 4 unique SS bond or disulfide bonds in native conformation. However, in one of his experimental conditions, you will see that there is a possibility of 105 different combinations and permutation of these 4 SS bond in the RNase molecule. I will explain how 105 combinations of 4 SS bonds are formed in RNase molecule. You have 8 cysteine residues which will be involved in the formation of first SS bond. What is the combinations or possibility? There are 7 possibilities or combinations possible for the formation of first SS bond. And once first SS bond has been formed, the remaining 6 cysteine residues will be involved in the formation of second SS bond in 5 different combinations. And after formation of second SS bond, the remaining 4 cysteine residues will be utilized in the formation of third SS bond in 3 combinations. After formation of third SS bond, the remaining two cysteine residues will be involved in the formation of fourth SS bond in one combination. So total combinations will be 7 times 5 times 3 times 1 is equal to 105 combinations of the 4 SS bonds in RNA's molecule. So you can say that there is possibility of 105 combinations of RNA's folded structures in one of his experimental conditions. So what is the significance of this in relation to his experiment? Remember two things. First, out of 105 structures, 104 are folded but inactive and unstable thermodynamically. So, and the second point is that only 1 out of 105 structures is in its active native conformation and is the most stable form. So next requirement is urea, which is commonly used as a denaturant in protein studies or in the protein folding. It disrupts all the non-covalent bonds and therefore all the second structure, tertiary structures will be collapsed or will be disrupted, leading to inactivation of the enzyme. Or you can say the enzyme will be denatured. However, it will not break the SS bond or covalent bond. So what will be the overall effect of urea on RNAs in this case? RNAs will be inactive. So denaturation causes inactivation of enzyme. Just remember that. So next requirement was beta mercaptoethanol. This is commonly used in protein structure and function studies. It is a reducing agent which will disrupt SS bond into free SH group. So what will be the overall impact on RNA's molecule? RNAs will be inactive. So alone beta mercaptoethanol will inactivate enzyme, alone urea will inactivate the enzyme, and both together, both urea and beta mercaptoethanol will also inactivate and denature the enzyme. Now coming to his experiment, Anfinsen conducted a series of 4 experiments. So in experiment number 1, he treated the RNA sample with urea and beta mercaptoethanol and the enzyme was denatured inactive, suggesting that enzyme is active only in folded structure and denaturation causes an activation of enzyme. In the second experiment, he tried to reverse denaturation by removing these components. So first he removed 8 molar urea followed by beta mercaptoethanol and this enzyme was active again, suggesting that denaturation is reversible. Enzyme is active only in folded structure or native conformation, and protein folding is a spontaneous reaction which is guided by the amino acid sequence of the protein. So in experiment number 3, he made slight change in the removal of these components. First he removed beta mercaptoethanol, allowing formation of SS bond in presence of 8 molar urea, and this is the condition where you expect to see 105 different folded conformation or 105 different SS bond formation in RNA's molecules. He then removed 8 molar urea which will not impact the SS bond already formed in this sample. So in this sample there is no urea, there is no beta mercaptoethanol. However, the enzyme activity was zero almost. Right? So what was the conclusion? The conclusion is that enzyme is active only in a uniquely folded native conformation. So all these 104 remaining structures, they are unstable, they are inactive, they are at higher energy level thermodynamically. So in experiment number 3, you have seen that although in the sample there was no urea, there was no beta mercaptoethanol, but the enzyme was inactive, although they were in folded conformation, right? So what he did, he tried to rectify by adding traces of beta mercaptoethanol in the 4th experiment. Beta mercaptoethanol will disrupt the incorrect SS bond in these conformations, and oxygen is there, so oxygen will enhance the formation of SS bond correctly. And because 104 out of 105 different structures, folded structure, they are unstable, they are thermodynamically unstable, they will easily be transformed into the native conformation. So this experiment suggested that the native conformation is the most stable and most favorable. That is why it is formed by traces of beta mercaptoethanol. So based on his experimental finding, he proposed that protein folding is a spontaneous reaction. Protein folding is guided by amino acid sequence of a polypeptide chain. This is very important point of his model, and thermodynamically native conformation is the most favorable and most stable structure.